Okay, that's it. Yeah. Abstract. It influences surroundings and also our emotions and behavior. We all have opinions about color. We have favorite colors and those that we don't like. Like today, I wanted to wear this red dress because I thought that I want to show up from the gray November landscape. Also, I think that red color is quite strong color. On the other hand, it means reminds me from passion and love, like what we have towards the research. And also, red color reminds me also from my PhD thesis, where I studied the anthraquinone compounds of the fungus Cortinarius sanguineus, the blood red webcap, and this fungus contains very bright red colorants, and therefore red color is also quite important for me. And it brings me to this research project that now we can start really researching biocolorants and try to find new solutions for colorants for the future. So we also know that colorants and coloration processes may cause problems to humans and environment, and therefore multidisciplinary research is needed to understand color and its meanings more profoundly, and to find new sustainable solutions to colorant production and applications for the future. Do you know what BioColour is? Have any of you heard about this project yet? There has been some Twitter accounts and some news on Aalto University's webpage or University of Helsinki webpage. No idea? Some of you have learned something. Okay, great. But hopefully today you can learn a little bit more of our project, and maybe you will also want to join our community and share and start creating new ideas with us. And today's program is about, you can see here, so Professor Harold Freeman from the North Carolina State University will tell us what color is about and how to produce it. This is the beginning of the color story. And then Ali Harlin from VTT and Package Heroes project will continue with colorants in biodegradable materials. And after that, then Professor Merja Penttilä from VTT will tell about colorants in the future by synthetic biology. And then we will have questions from the audience, so you can write down if there come some ideas during these speeches. Okay, what is BioColour? In our application, it meant bio-based dyes and pigments for color palette. So in this research project, which is financed by the Strategic Research Council at the Academy of Finland, and we have just started this project just in the end of June this year, and this project will last till 2025, so we will have plenty of time for cooperation. And there are 7 universities or research institutes involved in this study University of Helsinki Department of Education, Aalto University Arts and Aalto University Bio 2, University of Eastern Finland Kuopio and Joensuu, Häme University of Applied Sciences, Luke Natural Resources Institute Finland, and North Carolina State University, together with the University of Campinas, Brazil, and the VTT Technical Research Center of Finland. So we have very multidisciplinary group of people involved. And our aim is to develop new methods of biocolorants, large-scale production, characterization, and applications in order to produce degradable and safe dyes and pigments for textile packaging and coatings. And we also want to develop and foster biocolor entrepreneurship by examining social, cultural, economical, and environmental aspects of production and consuming, which able to build up novel processes leading to variety of sustainable items. And as you see in the middle of the picture, which explains our consortium, so we also want to gather all this information in this database, which can be used by all interested in the topic. And also what is important in this project is the impact and cooperation with different stakeholders and groups of people. So we want to build new knowledge with the scientific community, including, of course, the staffs, but also big resource are the students. We have business partners involved in this process, program, and also we want to communicate with consumers, and we are looking for participants for our consumer studies. Also, authorities and policy makers are in a role, and with those we do cooperation and discussions with the other financed research project in this in the Academy of Finland, and also for broad audience. So I'm a teacher by myself, and I think that the first-hand participation, personal experience and activity are the means to really get involved and to learn new things, and also sustainable ways of being in this world. So we want to create and foster biocolorant networks, orchestrate co-creation workshops where different partners develop solutions to material production and consumption. Also, we are going to arrange seminars and exhibitions which are open for everyone. So I hope that you look at calendars and, for example, at the university web pages, and we will pronounce our seminars and exhibitions there. And of course, we want to increase and disseminate knowledge of biocolorants and their processes, as well as sustainable consumption and lifestyles. So this is about our project, and we really want to invite everybody to join our community. We have also established Twitter, Instagram, and Facebook accounts, as well as our web pages will be in the address biocolor.fi. They are not the final ones yet. We will get the web pages launched at the end of November. And here are our also you can contact me or Riikka Alanko, our interaction coordinator. So let's go to our first guest. So I'm very proud to introduce Professor Harold Freeman. Harold was my opponent in my PhD thesis, so we have known each other for some years, but only with the email. And it was then this project came, and I decided that Harold must be involved with this.
Thank you.
So I'm very happy to have you here.
Thank you.
Floor is yours.
At my university, I teach for 75 minutes, but I only have about 15 minutes for this presentation. And so if you can find a seatbelt, you might want to fasten it for this part of the presentation. When we met, I'm sorry.
Excuse me, do you hear or should we take mics? Mic.
Okay. When our group started to meet, I guess a couple of months ago, we thought it good to cover this topic. Which we have been talking about biocolorants. Maybe this is better? Okay, good. You can tell I don't have a teacher's voice, even though I teach. A couple of my colleagues would never need a microphone as large as the classrooms are. But my pleasure is to talk about what colorants are. And this will be the perspective of an organic chemist or a color chemist, not a color physicist or color scientist. And so if you like organic chemistry, there'll be some parts of this presentation that you will really like. And if you don't like organic chemistry, then picture this as going into a museum, an art museum, and you see a picture and you look at it, you don't really know what it means, but it looks interesting, and you move on to something else. So think about organic chemistry experiences in this presentation in that way. Great. This slide shows the scope of the presentation. I'll start out with a working definition and then finish up by looking at the relationships between color and structure to show that this is not something that is mere happenstance. We can, in fact, predict and design the colors that we'd like to see in various molecules. We may not be able to do it as well as nature does it, but I think you'll find this an interesting point as well. I think Rika mentioned that this is a complicated matter. Even the definition of color is a complex one. And you'll be able to see that just by looking at a dictionary. This is a World Book Encyclopedia, and it shows 15 different definitions for color when used as a noun, and another 4 definitions for color when it's used as a verb. One of the things I pulled out of the first definition that was used were a number of words, and we'll see this unfold during the presentation, that light is a sensation. It's waves, or color is a sensation, I should say, and color involves waves, colors involve light. And color involves the eye. In fact, the eye is arguably the most significant part of of color itself. And the details that we see that I'll talk about in this presentation help us understand how it's possible for someone to have color vision problems. So color begins with light, and that's an important point. And if you think about what would happen if we turned out the lights in this room, we would have no appreciation for color at all. So for things to be visually colorful, there must be light. That is absolutely essential. And we see that the source can vary. It can be a lamp, it can be a fire in the fireplace, or at night, a fire, a campfire. But more often than not, the light source is the sun. And so it's white light that is responsible for color. And as this slide goes on to say, indicate that light moves in paths that we call waves. And we see in this diagram on the right-hand side that these waves can differ. Didn't mean to do that. Great, I got it. The waves can differ in terms of their diameter, the sharpness, or the broadness of these waves. And so we call these differences from peak to peak wavelengths. And so we learned that there are wavelengths associated with light, specifically as it pertains to light that we see in the, maybe I'll move over here because I'm a pointer when it comes to teaching. So when we look at what's known as the electromagnetic spectrum, just really a printout of waves of the different components of the electromagnetic spectrum, we can only see light in this region. It's known as visible light, and that's shown in the bottom part of the graph, so that there's no ability to see light to the right or to the left. So that's one interesting point. The second point that's interesting is that the part that we see is really a mixture of different colors. And we know that from something as simple as a rainbow. When a rainbow appears, this is really what we see. And when I was growing up, I always thought the primary colors were part of the rainbow. It wasn't until later on that I could appreciate that there were these various sections that not only showed red, green, and blue, but we could see orange and purple as well in most of the rainbows that we see today. Okay, so that's really the only time I need to be over there. And this is another reminder of just how incredible we are as humans. We don't have to do anything but just sit here, and we can see all of the colors that are represented in this room. But a lot takes place in the human eye in order to appreciate color. Light must enter the eye and move to the back of the eye in the sections known as rods and cones. And these rods and cones help us see at night and also help us differentiate between colors. And so the cones in particular are stimulated by light, the light that comes into the eye. It sends a signal to the optic nerve, and the optic nerve also sends a signal to our brain. And all of this takes place with one hand tied behind our back. We don't really have to do anything to cause that to happen, assuming that you don't suffer from color blindness. One of the things that's also interesting is that there are structures that are built into the cells in our eyes that undergo changes when they interact with light, and that also influences the colors that we see. Okay, so speeding up a bit, there are at least 4 molecular characteristics things that happen in order for us to see color. And that's a big deal because when you think of the hundreds of thousands of organic compounds that exist, less than 1% have color. So what's so special about that 1% that leads to the generation of yellow or orange or red or blue? And this slide really captures that. It shows that there must be absorption of visible light. If a substance can't absorb light in the visible spectrum, there will be no color. There must be conjugation, must be chromophores, and most dyes that are effective, certainly all of the ones that are commercially viable, will have oxychromes as well. So let's talk a little bit about that. And this slide shows that there is a relationship, a direct relationship between the color of light absorbed, that's the middle column, and the color that we see. And so for this surface that's on the right-hand part of the slide, that's orange, it means that it's orange because it's absorbing greenish blue light. If you put those two colors of light together again, you get white light. So that's an important point, that when light strikes an object, part of the wavelengths of light get absorbed by that object, especially if it's a colored object, and the remaining wavelengths are reflected from that surface to our eye and proceeds into our eye to the rods and cones that are in the back. Okay. The second property of all compounds that have color is this one of conjugation. And I know these chicken wire type structures may not mean anything, but conjugation, to the average person that is, there are alternating single bonds and double bonds. And you can see that with these structures. So that's a double bond, single bond, double bond, single bond, double bond, single. So this alternating sequence of single and double bonds represent conjugation, or we refer to that as conjugation. But the other thing that you see is that conjugation must be extended, though. So we need extended conjugation in order to have color. I didn't mention on the previous slide that the visible spectrum goes from 400 nanometers to around 700 nanometers. And so there must be absorption of light in that narrow window in order to have color. So the first two compounds you see will not have color, naphthalene and anthracene, but once we get to the tetracene compound, it absorbs light at 460 nanometers, and so it's an orange compound. Pentacene, the one at the bottom, has 5 rings. It's blue, absorbing light at 480 nanometers. This turns out to be a very expensive way to generate color, though, because pentacene, for 1 gram, and you probably put more than 1 gram, 3 times that amount in your tea or your coffee this morning if you take sugar in your coffee. And so that's not very much dye for $1,000. Just imagine having to pay $1,000 a gram in order to get color. The other point we see on this slide is that you don't have to have the major absorption band in the visible region in order to have color. A good example is vitamin A. Vitamin A gives this spectrum, so its principal absorption is in the ultraviolet region that we can't see, but it has this tail of an absorption in the visible region. And so this compound is yellow, gives weak yellow colors, versus the carotenoid at the bottom, beta-carotene, is orange. I'm not sure how I'm doing on time.
You can speed up a little bit.
Oh, okay. So we do need the seatbelts after all. The third point is that there must be a chromophore. This turns out to be an important point. I was afraid of that. It means that every compound will have some feature that's responsible for absorbing light in the visible spectrum. The dominant chromophore is the azo group, this nitrogen-nitrogen double bond, and second are anthraquinones, and many of the dyes that we'll be looking at in this project are anthraquinone dyes. This slide reminds us that The chromophore, though, must be part of a conjugated system. It cannot stand on its own. So we have colorless compound when the azo group is part of an unsaturated system. These methyl groups are CH3, but it's orange when you put it between benzene rings. So that's a big deal. The second thing we see at the bottom of the slide is that various groups can be attached to dye structures, and they are designed to do things like improve solubility, especially water solubility, give groups that can make ionic bonds to fibers, like the cationic nitrogen, but also a trifluoromethyl group that enhance brightness of the dye. Just one thing about azo dyes, since they're so important, these are synthetic compounds. We don't really know any that are commercially viable that occur in nature. They are a big deal, though, because they are tinctorially strong. You get a lot of color for a pound when you introduce these azo dyes, azo groups. They're easy to make. You can get them from yellow to black, so they cover the entire spectrum. However, some have this significant disadvantage that they were developed from genotoxic, that is mutagenic or carcinogenic aromatic amines. And this slide illustrates how azo dyes are made. I mentioned that they are simple to make. 2 steps, diazotization and coupling, and you get something like this. This is a structure of a highlighter, the yellow pen. That's how this structure is made. So diazotization and then coupling. It also means that we can look at an azo dye structure and know how it was made, so we can carry out a retrosynthetic procedure, start with the dye, break it down to the components that were used in the manufacturing of the dye. And that's important because our body has the ability to do that. The mammalian systems have azoreductase enzymes that can break down azo dyes to the constituent aromatic amines that were used in their manufacture. And that's a big deal for a dye like Direct Black 38 because one of its key precursors is this compound benzidine that causes bladder cancer in humans. So not a cancer suspect agent, it causes bladder cancer in humans. And so needless to say, this dye is not accepted in the marketplace anymore. And there are 20 other aromatic amines that fall into that category of banned from the marketplace. The right-hand side of the screen shows why these aromatic amines can be a problem, that they themselves, like the dyes, can be metabolized and they end up with species that will interact with DNA to cause mutations. Mutations are generally accepted to be the first step in the onset of tumor formation, malignant tumor formation. Okay, a bit about natural dyes. They have structures like this. Indigo is an example. This vat red 3 on the far side, and then the carotenoids like beta-carotene and lycopene. Also chlorophyll and various flavonoids that will probably be a part of this study as well. Natural dyes, though, even though we might wish this were the case, it's not the case. They were not designed with textiles in mind. They were developed with plants in mind to give us colorful roses, but not for textiles. So they don't have direct affinity some of them don't have direct affinity. So we have to develop good ways to bond them to the fiber. And that's part of what we're doing in this project at all. Many of them, some of them can be attached to cotton, and this as shown on the right-hand part of the screen, by using metallizing agents like aluminum and iron to form this mordantic complex between the dye and the fiber. Synthetic dyes, though, can be applied to the fiber in various ways by forming covalent bonds, ionic bonds, intermolecular forces of attraction like hydrogen bonds or dipole-dipole interactions or hydrogen van der Waals forces. But in this project, we're going to see solid-solid solutions like dispersed dyes on polyester and acetate and physical entrapment depending on the way these dyes are applied in our work. Here's some examples of natural dyes at the top, and at the bottom, synthetic dyes that look a great deal. So we're taking our cue from anthraquinone, synthetic anthraquinone dyes in our choice of fiber and dyeing conditions with the natural dyes we generate. One of the new methods we plan to use, or an additional method we plan to use, involves supercritical carbon dioxide as a medium. And so this means waterless dyeings is another potential opportunity for dye application. How much time? Okay, great. I mentioned at the outset that we can predict the spectral properties of dyes just by looking at their structures. And so once we know what we have and some of the structures we may need to characterize, we will know what their spectral properties are. And that will allow us to predict their reflectance properties as well as their absorption properties. That's the main message of this slide. Okay, in terms of a summary, what have we learned? We've learned that color begins with light. There must be a suitable source. There is this absorption and reflectance of light, and they are, I won't say a figment of our imagination, but they exist, they do exist in our mind in a very real way. We know the molecular requirements for substances that we isolate as biocolorants, the types of fibers they'll have affinity for, and we have the ability to predict, to know which ones to use in order to get a specific color. Lots of interesting books available to help us understand what color is. These are my 4 favorite, going back to my first book on color. I'm not sure that's commercially available still, but it's a fascinating little book. I'm also grateful to colleagues who helped me guide this presentation. My first inclination was to really make it all about organic chemistry, and they picked up on that and said, maybe you shouldn't do that. Let's talk about some other things as well. But thank you for your kindness.
Thank you, Harold. I'm sure we will have another opportunity to have maybe a little more longer lecture about color chemistry.
75 minutes.
Yes, 75 minutes next time. Okay, since Harold introduced some nice books, so I will introduce also some nice books. Because, of course, since you are here, you must be very interested about natural dyes. And me and Anja Primetta and Kirsi Niinimäki, we have written this book, Luonnonväriaineet in Finnish, and it's also translated in English, Dyes from Nature. And this explains some plant sources and fungal sources of dyes, algae, what kind of natural colorants they contain, also some dyeing recipes for those who want to do some experiments with dyeing or printing, and then also, which I think is the best part of this book, is the latter part, and it explains about the dyeing properties and the textile chemistry and textile technology part of natural dyes, what kind of properties they have and how they attach to the fibers. So these books are available from the internet bookstores. So if you are interested in, you can now go and buy yourself an own one, or they are also found from Kaisa Library. Okay, and if you are interested to see, so the book is available here. But our next speaker is Ali, here. Ali Harlin. No Ali around. Okay, busy man. But no problem, maybe he will come and we can now then go to Merja's presentation. Okay, so yes, I will give this. Wait a minute. Now we need this.
I can take this.
Yes, yes, but we need to both because this goes for the tape, for the recording.
Okay.
I will have this. Oh, maybe it can be there. There.
Fine?
Yes.
Okay. And I'm also actually in the program you have that there should have been a video from Leslie Mitchell from New York State University, but actually we did not get that in time, so I try to also cover a little bit of what Leslie would have been saying. So I talk about synthetic biology in relation to color, and I first want to make quite a bold statement that synthetic biology will enable in the future the production of any biocolor in simple microbes. So you can take the genes from whatever organism and put them into microbes, and that you can then also produce at industrial scale. So this is our wish, and there is quite a lot of activity at the moment towards this goal. And here are some examples on what microbes themselves, because they do naturally also colors. You don't need to take the genes from other organisms. You can see that here is, for instance, a yeast, which the colonies, yeast colonies on a plate, and they are reddish. You can have bacteria which produce violet color. You can also produce with bacteria, you can produce indigo, the blue color, which plants can do. And then you have various different fungi here, penicillium, for instance, which can produce a very strong reddish color. And here is another fungus, Fusarium, on a plate, and you can have them producing different blues, reds, yellows, and so on. And these then, in a way, the fungi, the filamentous fungi, which are growing in the plate, so sometimes they then make these type of structures which you recognize. So actually the mycelium of the fungus is growing underground, and it's very similar to what you have of these fungi on the plate there. But then they make these fruiting bodies which we know as mushrooms. And of course this one is very colorful. And this is, I guess, Riikka's favorite mushroom, producing a very pink anthraquinone color. And this one, of course, has been used as a source for natural pigments in traditional handicraft, making handicrafts. So why do microbes produce pigments? So this is, of course, we do not know, but there could be photoactivity. They could be the sort of simple eyes of an organism, so they perceive light. Also, they could be used as energy absorption, so that the organism gets a little bit light energy while the light hits the pigments. UV protection, like algae or hallow-tolerant bacteria, which are living in seas. So they see a lot of light, so they need to protect themselves. They could be antioxidants, or and protection against oxidative stress. And then, of course, they could be attraction, like the mushrooms attracting the insects to come and then spread this way their spores to different environments, other environments, or then they could be there for protection. So they could have antimicrobial properties. And of course, these could be something we can also consider, that while they produce as color, they may be also somewhat useful in other terms as well. Usually these properties are not essential in laboratory where we grow the microbes, so we need to boost up, for instance, use twinkling with the genes so that the color becomes then visible for us. So here are some examples, and we saw them already in the previous presentation, of different kinds of molecules which then produce different colors, or different molecules, pigments, which then produce or are perceived as different colors. So you see the same structures here. And to say that the microbes also produce similar type of molecules than plants, for instance, produce. So we have a rich, there are variety in nature, but there is also very much similarity in nature. And this is important to also remember, because then you can start playing around. I mean, it's a common feature for all organisms that the structures are similar, and maybe also the genes behind are similar. This is another example where we have aromatic compounds, and some of you may be seeing the rich sort of biochemistry now, what the microbes have. So you can start here. This is a sort of a cell. You can start the cell taking up sugars, and then the sugars are taken in the metabolism of the organism to these different structures. Here is indigo, here is violacane, here is pinosylvin, and here is, for instance, gallic acid, as examples of colorful compounds. And here also you see the chemical structures, but you also see the enzymes which do the biosynthesis, and also you see the genes behind the enzymes. So nowadays many of the genes are known which are coding for these reactions. Of course, there is still lots to discover, but in principle we can now start to do genetic engineering and synthetic biology to play around with the pigment synthesis pathways. And this comes in, we can come then to synthetic biology. I don't know how many of you have heard about it. It is sort of an upgraded version of genetic engineering, where you take the genes and transfer them between organisms. But now really synthetic biology is revolutionizing biotechnology. And why is it so? It's because we have so large genome data sets now. Like in this BioColour project, we could actually start sequencing the genomes of the fungi. It wouldn't cost so much that we could do that, and then go and try to see which of the genes in the genomes would code for which kind of color compounds. So what is the possibility of certain microbe to produce a certain type of a color? Now then also we can have very cheaply, we can already synthesize DNA, long stretches, in vitro, not in a living organism. And that means that we can also design how we want that to be. And we can make big pieces. We can take several genes, we can modify the genes, and then we can order those genes from a shop. And then we can put them into the organism we want. And then the biosynthesis pathways would then be occurring through similar reactions as we humans have also, how the genetic codes works and how the biosynthetic pathways in any living organism work. So we can design and build cells as we wish. And then we can also apply different principles of engineering sciences nowadays in making this in synthetic biology. So it becomes much more easy and much more broad. In a way, we can broaden the diversity we are doing, and it comes much more fast and cheap to do this. So this is the concept, in a way, biotechnology, so that we have a raw material, whatever it is. You know that fungi molds can grow on anything, waste material, or algae can grow on CO2, carbon dioxide. So you take the raw material in, the biosynthetic pathways take care if we have engineered the microbe, and then you get a product. It could be a fuel, a chemical, a material, but it could also be a pigment. And in comparison to chemistry, like fossil chemistry, we can now tailor the microorganism so that it makes only the wanted product, like the pigment, and as efficiently as possible. And then we can grow the microbe in bioreactors, so it's contained, so it doesn't escape to the nature. It's a sort of a single unit operation when you just give the microbe the raw material, like sugar. It takes care of long metabolic pathways, biosynthetic pathways by itself, and makes the product. So you don't need to do, as in chemistry, that you usually do one reaction at a time, and then you purify the product, and then you go to your next one. So this is the biosynthetic power of living organisms, that they do it dictated by their DNA, by their genes, and the power of life, in a way. And we can do this in room temperatures or ambient temperatures, no toxic catalyst, and no high pressures. And then when we sort of, on top of that then put the novel aspects, what synthetic biology brings in, so we can use computer-aided design of the metabolic pathways, of the synthetic pathways, of the whole organism if we want. How do we want that to perform, what we want that to make. And we can also nowadays use automation, robotics and so on to put the DNAs in, to select from hundreds of strains the ones which are performing as we wish. And we can have new reactions, not only the ones we find in nature, but we can also fine-tune the reactions. We can have new products, and we can also have more efficient processes. So this is now the sort of a promise of synthetic biology. And this is now the part which we wanted Leslie Mitchell to talk about because she has made a nice study which relates to actually pigment formation. So she has taken genes which are responsible for carotenoid production and also a few other genes, and now then wanting to make a baker's yeast, which we know that it doesn't produce pigments, and it looks on a plate white, and to now construct pigment producing pathways into the yeast. And so she took these genes and made it in a combinatorial way, so different genes in combination to get maybe different modification of the complex chemical structures which we saw, and which would then give a different color. And so here you then see what is obtained. So the different individual yeast strains, which are now here making colonies, so they are starting to produce different colors, and then you can nicely purify also the individuals, and you can then sequence and see which of the biosynthetic genes that particular yeast is carrying, which makes a particular color. And what was also cool, and this is the sort of what happens in synthetic biology, that the students are very, very inspired about various things. I mean, in particular, young students. And I think this was also partly a student project, that they wanted now to play around with the different yeast strains, which are making the different pigments here, and then starting to reproduce an image, which they then sort of had all the sort of pigment characteristics or specifications, the indexes, in a way known what this picture or any picture has, and then they looked for the corresponding color, and then they had an automated program and a robotic system which then took that particular yeast into a specific position so that it actually reproduced the picture. So you can see here that there are dots of the yeast here growing. So making these kind of living pictures, and these are they are also selling, so if you would like to buy one. And this just shows that what synthetic biology biologists are sort of playing around with, that they can make living pictures and now use the yeast strains producing the pigment. Of course, we would like to go beyond that, and this some of you may have seen that we have been producing these Korva headphones with the design company Ivan, and these actually contained around 5 different materials, and they are actually all produced by microbes. So there is, for instance, the bioplastic PLA, which is something which you have in most of the it's cups, and it's the most sort of prominent bioplastic here, and we can make that with yeast. Then there is cellulose made enzymatically or by bacteria. There is also fungal mycelium, which is made to a leather-like material. And then there is also biosynthetic spider silk. So we have taken the genes from spiders, put them into a microbe, and that produces the silk. And then there are some other things. So of course now, even though these materials are not really ready to be sort of for mass production, but I mean this just is a vision in a way that we can start also producing the materials with microbes, and then also it would be very nice and cool if we could also produce the pigments to color these materials with microbes and with synthetic biology. And this is now what we will be doing also in the BioColour project. And just to sort of finish that, it is so that even though, like this, here is now the baker's yeast and here is a mold, so they are very tiny and you can fit into a small, like 1 liter bioreactor, like at least 10 to 8 individual cells, so they can really be taken also to large-scale production. So if we would like to produce the pigments at industrial scale, so in principle that would be possible. And I sort of, it's of course, nothing is sustainable by itself, or even if it would be biotechnology or natural, it wouldn't be necessarily sustainable. But I would think that we can foresee that microbial production of pigments would be sustainable, because you can use renewable raw materials, plant waste fractions, or even directly carbon dioxide as raw materials in the production. We can boost up the production so that we can really increase the yield of the product, and we can produce also a pure compound if we engineer the cells to do that. So we get a very little of waste, or less waste than with other means. We can get the production in enclosed bioreactors, as I said before, but importantly also all year round. We are not dependent on the, I mean, you just have the organism, you put it in a bioreactor and it produces. And we have no need to extract the pigments from, let's say, natural plants. Of course, in some cases, and it's studied also in BioColour, that's probably the most reasonable thing to do. But in principle, we don't need to extract them from plants. From poisonous mushrooms, we just take the pigment synthetic pathways, or insects, like my lipstick is from Lanzarote, from this carminic acid which the coccinelle are producing. And there are no land use issues, because you just have the bioreactor where the production is occurring. And so, still to advertise the book, Riikka's and others' book, and then also to say that, which I usually say after my talks, that biology is really inspiring. So I think we have lots of solutions in nature, which we need to now sort of take into use.
Okay, thank you, Merja. Okay, I just learned that Ali had a meeting somewhere too far away and he didn't make it here, but I'm very happy that we are good in a schedule. So now there will be, it was planned for a half an hour questions from the audience, and we have a real nice audience here, and I'm sure that you will have questions, will you? So if you have any ideas or any things in your mind, what did you think about this synthetic biology? Have you heard about it before? Does it raise any questions?
One question. When you talk about these pigments to color, is it pigment or is it dye? Is it actually a pigment?
Yes, the questions about pigment and dye. This is a very good question, because when we are in a field of natural colorants, we come to the question that in different disciplines, these molecules are called in different names. And in biology, these natural compounds are called pigments. But when we look at it from the coloration point of view, so pigments are not water-soluble compounds, whereas dyes are water-soluble compounds. So in color chemistry, there is difference between dye and pigment, but in biology, all these natural compounds are called pigments. So these in biology, these pigments are water soluble. Yes. So this is a little bit confusing.
Yeah, that's true.
Yeah.
Now I see what you mean by biopigment.
And we even, I recognize I had to kind of teach a little bit of these terms to my colleagues, which haven't been so much involved with this color chemistry. What word? Because in Finland, in Finnish, we have only this väri, and this is for everything. And in English, we have these different words, and I even get and mixed up. I have to be careful what words I use. But any other questions? Yeah, Gisela.
I was wondering how you can get the dye from that biomass, and also thinking about natural. We have a lot of toxins in natural world, so how you propose to do it? You test it before the thing you want, or how you know that the byproducts are being produced?
Well, I guess the byproducts we can sort of analyze, of course, and then we can think about whether we can knock out some side product genes to reduce the byproduct formation. Of course, it may be rather complex, we know from the pigment synthetic pathways. Then, of course, in principle, I mean, if we don't do anything special, so the pigments need to be extracted from the fungus, the microbe, similarly as it's done, has been done before. And we know that some of them come naturally to the medium, some may stay inside the cell, and whether it's then the sugar additions or other features of the molecule which affect whether they come to the medium or not. So we know that there are different pigments or compounds in this respect. Of course, with lots of research, we could also analyze whether we can have some ways to export the molecule out of the cell, because this is something which living cells do also if there is a sort of compound they don't like. So they may find a way to have molecules in the cell membrane which can aid in exporting the molecule out. But this, I guess, is something we do know very little at the moment yet.
Great. More questions?
Merja, the question is about the reason, of course, why there's so much interest now in biocolors, which we haven't seen in so many decades, is because of toxicity of the non-natural coloration processes. So you really believe this will be the solution to get rid of massive pollution of the dye processes that we have now in, for instance, the textile industries?
At least we can have, in principle, at least nicer processes, because we don't need to use the many different chemicals, and some of them are nasty chemicals in the dyeing processes. But of course, some other people know here better of that. And of course, some of the natural pigments may also be toxic. So this is one of the things which is being studied in this project as well. And Gisela is here somewhere. Yeah, there you are. So you may comment on this. And we know that there are sort of modifications of the molecules which make them non-toxic or toxic.
Yeah, I can. Yeah. Go ahead. Okay. And I can also answer that. Of course, there are lots of different like types of synthetic dyes and lots of different applications. And we won't say that we create solutions for every application. It is that we can do something and we can start something new, and maybe in some time they can be in application in some places, but at least it's our opportunity now to create more sustainable alternatives for some applications at the moment, not all. But I don't know where this world is going. One target in our research project is also to study the color as a psychological phenomenon, and also what is actually the color, and what do we mean about it, and how it affects to us, because this might be that in the future we need to kind of think and rethink about what is color and what we mean about it, if we want to act and live in a sustainable way.
And is it reduced to human perception, or are you looking at other entities, how they perceive color? For example, develop some color that the mosquitoes don't like? Just saying something very practical.
maybe we are in the human world at the beginning. But Gisela, do you want to add something about this toxicity?
Well, I think we need to see the broader picture. It's not only the dye, because you need to think about a little bit about risk assessment and not about only hazard. People pursue this non-toxic thing, and as a toxicologist, this doesn't make any sense to me. Of course, we want less toxic compounds, but we need to see the whole picture, thinking about humans, dermal exposure, environmental, how much this is going to wastewater. So those processes, if we analyze in a perception of the cycle, the whole cycle of the production, then we can have a nice view. So I don't think we need to get rid of everything, but maybe just being smarter, right?
Yeah. And I want to add also that about this, what is toxic, is about a question of dose also. If we read about articles about coffee or red wine, it is just always comes to the question, how much do we drink it, if it's good or bad.
One comment, and when I said that I think in the future we can produce any BioColour or biopigment, yes, but it doesn't say anything whether they are toxic or not. So just in a way that you can do these things nowadays, and it's a little bit the same as with organic food or non-organic food, because I mean, molecules are toxic or not toxic, and it depends, as you said, on the dose and so on. But at least there are certain reasons, as we have discussed, why we think that we could produce something which would be more sustainable.
Yeah, more questions? Yep.
Thanks all for inspiring talk. So I have a question about these bio dyes or biological processes. So how do the embedded energy compares with the synthetic processes of synthetic dyes, or is there that kind of justification that you can also produce them in much more like energy efficient ways?
So this is something we are going to study in the process, because we also have techno-economic analysis and LCA analysis, life cycle analysis. So of course we do not necessarily know yet, but let's say that at least sort of in general, if we think about production with biotechnology, if we for instance take the case of PLA, the bioplastic, so we know that if we look at the CO2 footprint, for instance, that's much more lower than if we take the fossil bioplastics of different types. So by using cheap raw materials, often waste fractions as raw material, then having this single step process, reduces, let's say, energy costs, and also but then what we need to take into account is that biotechnology uses also quite a lot of water, and also then we need to take into account the downstream processing, the purification of the products. But at least the downstream processing, of course, is similar, I guess, to the synthetic chemical dyes. So one should then dissect in a way all the steps and see what their energy and CO2 impact is. But there are, I mean, I listed certain things there, so there's a good sort of starting point to think that one could get a sustainable process this way. But this we need to analyze.
I am wondering if there is a study if, let's say, a person is wearing biocolors like all the time, that's some kind of long exposure to the skin, does it have any effects or not? Maybe like in the future would be the clothing is something that everybody wears biocolors. So.
We have made some preliminary studies with some cell tests about dyed textiles, and those didn't show toxicity in the tests. When we dye in a traditional way, the compounds are mixtures of compounds that are in the textile, and also the amount that attaches to the fiber were very low. So also then in the tests what we make now in this project, we will test pure compounds. So we will extract pure compounds from those mixtures and study then the toxicity of those compounds. Then it's more straightforward also to consider what compound shows toxic activity and what not.
So the study is about toxic, but any other positive effect is not studied?
There are some natural compounds that are considered toxic and pseudotoxic, and some other, like some other, I don't know quite the terms how you call them, but show that they are not safe. But then there are also compounds that doesn't show that. And of course, if we test the textiles, it also showed that since the textile contain a lot of different treatments, it is also difficult to or is it's also important to test the textiles themselves, what compounds they contain.
Maybe I can just add a comment. Well, we have tests for that, for depending on where you are using the dye. So if it's to eat as a food thing, or if it's going to be textile, but we have protocols to test, and it doesn't matter if it's a BioColour or it's a synthetic one. We're going to do the same tests in order to approve it. So it's not because it's natural that we're going to say, but it's the whole idea that I think it's interesting to see. But we follow procedures. Okay.
Any other questions? Okay. If there are not any questions, I want to thank you for coming, and I hope that you will then enjoy some drinks with us, and hopefully we will see and keep in touch And if you are interested, then you will find our details from the internet. Thank you.